Pathogens, before successfully establishing an infection, must first manage to enter our body. There are many portals of entry for pathogens, including the skin, the mucosa of the respiratory tract, the gastrointestinal tract, and the urogenital tract. Pathogens could also enter through the eyes, ears, nose, and oral cavity, and also through glandular tissue such as the lacrimal and the salivary glands. Pathogens could also be introduced to the body during a diagnostic or therapeutic procedure, such as using an IV cannula or using a urinary catheter, which creates potential routes for microbial invasion. Now, the surfaces or portals of entry for pathogens represent the anatomic epithelial barriers, which insulate and protect the body's interior and can effectively resist the invasion of most pathogens. Epithelia in these locations are specialized for their particular functions, and they possess unique innate defense strategies against the microbes they typically encounter. Now, the three main surfaces in our body that are heavily exposed to pathogens are the skin, the respiratory tract, and the gastrointestinal tract. These surfaces are in continuous contact with pathogens. Out of these three surfaces, the most resistive and at the same time the least exposed to the external environment is the skin. Since skin consists of several cell layers, it is very resistive, and there are only a few microorganisms that are able to penetrate intact skin. Now, in the respiratory and intestinal mucosal tissues, we don't see such layered epithelium. Instead, there's a single layer of epithelium that covers these two tissues. The respiratory epithelium, although looks stratified, it actually consists of a single layer of cells that are staggered in a way that suggests multiple layers. The gastrointestinal epithelium is also composed of a single layer of cells that covers our villi. Now, due to this single-layered epithelium in our airways and gut, the mechanical barrier that is offered here is much less resistive than that of the skin, and it is relatively easy for microorganisms to pass through these more fragile mucous membranes. And the reason why they are only a single cell layer thick is to facilitate nutrient uptake in the intestine and to facilitate gas exchange in the lung. Now, that makes sense that skin is the most resistive due to the numerous layers of the epidermis. But how is it that skin is the least exposed of the three to the external environment? To answer this question, we must first know the surface area of these three tissues. Skin has a surface area of about 1.7 square meters, whereas our respiratory epithelium has a surface area of about 70 square meters. And that is the largest area of contact with the microbial environment among all tissues. The gastrointestinal tract is also heavily exposed to microbes and covers a surface area of approximately 32 square meters. So although skin is the largest organ of our body and accounts for about 15% of total body weight, it has a very small surface area compared to the enormous surface area of our mucosal tissues that are exposed to the external environment. So the skin is the most resistive and the least exposed, whereas the respiratory system is the least resistive and the most exposed. The lower respiratory tract is least resistive because of its fragile mucosa that consists of only a single layer of epithelium. And it is most exposed not only due to the extensive surface area, but also due to the high rate of continual air exchange with the environment. Our average respiratory rate in a minute is around 12 to 20. Let's say it's around 14 breaths per minute. Multiply that by 60 minutes, then by 24 hours, and that would sum up to around 20,000 breaths per day. Each breath we take, we introduce about 500 milliliters of air into our lungs, and that is called the tidal volume. So here we're talking about 10,000 liters of air entering our lungs on a daily basis. And that's a huge amount that is enough to fill up an average-sized swimming pool. Additionally, we don't have a good competing microbiota in our lungs, such as that of the gastrointestinal tract. These three factors make the respiratory tract a major portal for the entry of pathogens, and that explains why respiratory infection is the most common infectious disease. Now, in accord with the large surface area of tissues exposed to the external environment, there has to be efficient immune defense strategies that protect us from invading pathogens at those surfaces. And the first immune defenses employed by our body at those surfaces are called the barrier defenses. These barriers consist of physical, chemical, and biological barriers to infection, as well as mechanical defenses that aid in pathogen removal. These four properties of our epithelial surfaces are key elements of innate immunity, and they actually constitute the innate immune system's first line of defense against infection. Barrier defenses are not a response to infection, but they are continuously working to protect against a broad range of pathogens. And they tend to deal with pathogens in a strategy that involves mainly avoidance. Avoidance is a mechanism that prevents exposure of the host's internal tissues to microbes in the first place, and this strategy is widely used by our barrier tissues. Now, if these barriers are breached and an infection is established, then another strategy called resistance will get involved, and its purpose is to reduce or eliminate the invading pathogens. So the innate immune system's first line of defense include these four main barriers that prevent microbial invasion, and we'll discuss each one of them in details, beginning with the physical barriers. All tissues in our body are lined by a strong barrier of epithelial cells joined together by tight junctions. This epithelia that lines the barrier surfaces could be made up of multiple layers of cells, such as that of the skin and the reproductive and urinary tracts, or could be made up of a single layer of cells, such as that of the intestinal and the lower respiratory tracts. Now, the epithelial cells in these tissues are joined together by tight junctions. These tight junctions are actually a protein complex that stitches these cells together and gives these surfaces the physical properties that effectively form an impermeable seal which prevents molecules and pathogens from squeezing into the intercellular space. Now, the most effective physical barrier out of these surfaces is our skin. The skin's epithelial layers and tight junctions between epithelial cells are like a shield that seal us against the external environment. Skin is considered an extremely efficient physical barrier, as most pathogens cannot invade intact skin. The majority of pathogens gain entry through the skin if it is damaged, and a common example is skin wounds. Skin consists of two distinct layers, a thin outer layer called the epidermis and a thick inner layer called the dermis. These two layers are separated by a basement membrane. The epidermis contains lots of layers of tightly packed epithelial cells, and the outer part of it is called the stratum corneum, and it consists of about 15 layers of flattened dead cells called corneocytes. These corneocytes have lost their nuclei and all their organelles, and are instead filled with a waterproofing protein called keratin. Corneocytes are enclosed by a thick, mechanically stable and highly insoluble protein-lipid envelope called the cornified envelope, and also an additional lipid envelope that replaces the plasma membrane of living cells. The structure of corneocytes with keratin and the envelopes makes the skin's surface mechanically tough and resistant to degradation by bacterial enzymes, and also helps to make the outer surface of the skin relatively waterproof. And that helps keep the surface of the skin dry, which is very helpful in reducing the microbial growth. In addition, these corneocytes are tightly linked to each other by adhesion protein bridges called corneodesmosomes, which constitute the principal mechanical junctions that reinforce the uppermost layer of our skin and gives it its physical strength. Another example of a physical barrier that is common to mucosal surfaces is mucus. Mucus is a viscous fluid secreted by specialized cells of the mucosal epithelium called goblet cells. Goblet cells can be found in various mucosal tissue but is most abundant in the lining of the intestine and the respiratory tract, where mucus is essential for normal function of those organs. Goblet cells manufacture a glycoprotein known as mucinogen, which when released into an aqueous environment becomes hydrated and is known as mucin. Once the mucin is mixed with other material in the watery environment, it is now known as mucus. This mucus, in addition to its lubricating function, acts as a physical barrier, which covers and protects the more fragile epithelial cells beneath it. Mucus impedes microbial access to various epithelial surfaces by entrapping them and inhibiting their movement further towards the epithelium, and eventually preparing for their expulsion out of the body. Now in the lung, one of the first lines of defense is the airway surface liquid, which lines the airways. The airway surface liquid is a fluid layer covering the airway surface at the interface between surface epithelial cells and the airspace. The airway surface liquid comprises two layers, a mucus layer contacting the air, which overlies a periciliary layer that bathes surface cilia and contacts the surface of epithelial cells lining the airways. Both the mucus layer and the periciliary layer contain a complex mixture of soluble and bound mucins, which increases their viscosities much greater than that of water. Mucins of the mucus layer are secreted mucins that form a continuous gel-like lining, whereas mucins of the periciliary layer are tethered mucins that are bound to the epithelial surface, forming a glycocalyx lining. The tethered mucins of the glycocalyx not only pose a barrier to the penetration of the periciliary layer, but may also confer a lubricating property to facilitate ciliary beating and to ensure effective mucus transport. The periciliary layer is separated from the mucus layer on top, and it is also much less viscous than the mucus layer, and that is very important for the movement of the cilia that it surrounds. The upper airways are characterized by a thick mucosal layer, which is rich in goblet cells. Goblet cells become less numerous as the airway becomes smaller, so the lower airways, specifically the terminal bronchioles and the alveoli, lack goblet cells, and as a result they lack the thicker mucosal layer characteristic of the upper respiratory tract, because efficient gas diffusion would be impeded by a mucus layer covering the alveolar epithelium. In contrast, the mucus of the intestine becomes thicker as we go down towards the colon, or in other words, the goblet cells becomes more numerous as the villi becomes shorter. In the small intestine, both goblet cells and Paneth cells are responsible for mucus production, but create a much thinner layer than that of the colon. The mucus layer is thin and focally discontinuous in the proximal small intestine, becoming thicker and continuous in the ileum. These focally discontinuous areas that are devoid of goblet cells and not covered by mucus are the follicle-associated epithelium, which is associated with the Peyer's patches and the isolated lymphoid follicles, and which is designed to facilitate sampling of the luminal contents. In the large intestine, the mucus layer becomes very thick, correlating with the density of goblet cells in the different regions of the intestine, which generally has an inverse relationship with the length of villi and a direct relationship with the abundance of commensal bacteria. So the density of both goblet cells and bacteria is greatest in the large intestine, and the density of each declines proximally. The mucus of the large intestine actually is composed of two distinct layers. An outer layer, which is a thick and porous layer, in which commensal bacteria can reside, and an inner layer, which is thin, densely cross-linked and attached to the surface epithelium, and that layer is generally devoid of microbes. This inner layer is very dense in its structure and limits bacterial penetration by having pore sizes smaller than bacteria, and that maintains a healthy distance between epithelial cell surfaces and the commensal microbiota, creating a zone that is almost completely free from microbes. So in summary, mucus increases as we go up in the respiratory tract towards the upper airways, and also increases as we go down in the gastrointestinal tract towards the colon. Next, we have the chemical barriers that block entry of pathogens on the outer body surfaces, at body openings, and on inner body linings. So all of these surfaces, in addition to their physical barrier properties, also have specialized chemical products that can either kill pathogens or weaken their effect and make it difficult for them to survive. The chemical barriers include mainly the antimicrobial proteins and peptides and the acidic pH. Antimicrobial proteins and peptides are referred to as AMPs, and they are multifunctional defense molecules that provide passive protection against infection. And besides their antimicrobial activity, they also modulate the immune system by activating immune cells against pathogens. AMPs are mainly found in human body fluids or secretions. Additionally, they can be found in some cells and their secretory granules, and can also be produced by some of our commensal microbes. Example on a natural antimicrobial that is produced by all barrier surfaces include the family of amphipathic peptides called Defensins. In humans, there are two classes of defensins, alpha defensins and beta defensins. Beta defensins are produced nearly by all barrier epithelium, whereas alpha defensins are produced by Paneth cells in the intestine and by neutrophils and macrophages. These antimicrobial peptides act as a chemical barrier and protect the epithelial surface from different sorts of pathogens. Defensins kill pathogens basically by disrupting their cell membranes and by creating pores that make their membranes leaky. Another example on an antimicrobial that attack chemical features specific to bacterial cell walls is the antimicrobial enzyme Lysozyme. Lysozyme is an enzyme found mainly in body secretions, such as saliva, tears, breast milk, mucus, and cervical secretions. Lysozyme is very effective in acting against gram-positive bacteria because it can cleave the exposed peptidoglycan component of their cell walls, leading to their digestion and lysis. Now, the other example on a chemical barrier is the acidity of some of our tissue surfaces. The low pH environment caused by acid production protects many tissues of the body, and due to that acidity, lots of pathogens are either killed or inactivated. Acidic environment in our body include mainly the stomach, the vagina, the ear canal, the skin, and the urinary system. The stomach is an extremely acidic environment due to the hydrochloric acid in the gastric juice, which breaks down food and kills many ingested microbes. In addition to the acidic pH of the stomach, the digestive enzymes, bile salts, fatty acids, and lysolipids present in the upper gastrointestinal tract create a substantial chemical barrier to infection. The female genital tract is also acidic, and that's very important in providing protection against bacterial and fungal pathogens in the vagina. The external auditory canal is slightly acidic with a pH value of about 5, and that plays an important role in inhibiting bacterial growth. Additionally, cerumen also exhibits antimicrobial properties due to the presence of fatty acids, which also contributes to the low pH in the ear canal. The skin is also slightly acidic with a pH of around 5.5, and most bacteria fail to survive for long on the skin because of the direct inhibitory effect of lactic acid and fatty acids in sweat and sebaceous secretions. In the urinary system, the slight acidity of urine inhibits the growth of many microbes and potential pathogens in the urinary tract. Now, the mechanisms of maintaining this acidic environment are numerous and different in each location and are beyond this lecture. But there's one mechanism that you must know because it's very important clinically, and that is the acidity of the female genital tract. If we take a closer look at a vaginal epithelial cell, we'll see little rod-shaped flora called lactobacilli. Lactobacilli is a gram-positive bacteria that typically colonize the vagina and cervix and maintains an acidic vaginal microenvironment. The vaginal epithelium produce and accumulate glycogen, and under optimal conditions, lactobacilli consume this glycogen and metabolize it, resulting in lactic acid production. That's why it's called lactobacilli, because it's a lactic acid producing bacilli, and this lactic acid is what maintains the acidity of the vagina to less than 4.5, and that's what inhibits transient microbiota, opportunistic pathogens, and other pathogens responsible for sexually associated infections. Now, if an imbalance in vaginal flora occurs, like a decrease in lactobacilli colonization, this will result in an increase in the pH and loss of the protective acidic vaginal microenvironment. And this leads to an overgrowth of anaerobic bacteria, such as Gardnerella vaginalis, which causes the sexually associated infection bacterial vaginosis. Bacterial vaginosis is the most common vaginal infection among premenopausal women and could be diagnosed by the AMSEL criteria, which includes a thin white vaginal discharge, a fishy odor upon doing the whiff test, presence of clue cells coated with bacterial biofilm, and an elevated pH of more than 4.5. Next, we have the biological barrier. Each of the epithelial surfaces harbors communities of microorganisms that coexist in a healthy human body. We have trillions of bacteria, viruses, protozoa, fungi, and even worms sharing our human bodies and living in relative harmony at our barrier surfaces. Collectively, these resident microorganisms are referred to as commensal microbiome, and they constitute a biological barrier to infection. The term commensal means eating at the same table, and commensalism refers to a symbiotic relationship between two organisms where one organism benefits and the other derives neither benefit nor harm. But that's not the case here. Although these friendly microbes are usually referred to as commensal, research in this field suggests that the relationship between our microbiota and us is not merely commensal, but rather a mutualistic relationship in which both species benefit. This microbiota uses a variety of mechanisms, and it competes with pathogens for cellular binding sites and nutrients, making it hard for them to settle and penetrate our body. The commensal microbiota is very unique in the gut, especially in the large intestine. The large intestine is the site of the most abundant and the most diverse microbial community in the body. As we mentioned earlier, the large intestinal epithelium is covered with two distinct layers of mucus. The outer layer is a more porous layer in which commensal bacteria can reside, whereas the inner layer is a densely cross-linked layer which has pores smaller than bacteria, and that maintains an additional boundary and a healthy distance between the microbiome and the cell surfaces. The importance of this normal microbiota in host defenses is highlighted by the increased susceptibility to infectious diseases when the microbiota is disrupted or eliminated. For example, when we use certain broad-spectrum antibiotic treatments, occasionally we may end up in killing our beneficial commensal microorganisms. Now, if this normal microbiota is significantly depleted, we may have superinfections or overgrowth by antibiotic-resistant opportunistic microbiota, such as Clostridium difficile. Clostridium difficile is a spore-forming, gram-positive, anaerobic bacteria that produces toxins which cause severe bloody diarrhea and intestinal mucosal injury, and could result in the disease called pseudomembranous colitis. One strategy for treating recurrent antibiotic-resistant Clostridium difficile infections is fecal microbiota transplantation, which involves the transfer of fecal material from a donor into the intestines of the recipient patient as a method of restoring the normal microbiota and combating Clostridium difficile infections. Finally, the mechanical and physiological defenses. We have a number of mechanical defenses that are essential in removing pathogens away from the body before they manage to establish an infection. So when you hear the words removing, shedding, expelling, excreting, or flushing of microbes, all these imply the mechanical removal of microbes from potential sites of infection. For example, the shedding of dead skin cells on the surface of the epidermis takes away with it many microbes. Perspiration from our sweat glands also flushes microbes from the skin's surface. The expulsion of mucus by the coordinated beating of the cilia via the mucociliary escalator drives the mucus-entrapped pathogens up to the nose and mouth, where they can be either sneezed or coughed out of the body, or could also be swallowed into the digestive tract, ending up in the harsh acidic environment of the stomach. The excretion of feces from the rectum by the help of intestinal peristalsis expels lots of microbes carried in stool. The flushing action of urine washes microbes out of the body and is largely responsible for the normally sterile environment of the urinary tract. The flushing of tears with the aid of the mechanical action of blinking serve to wash microbes and debris away from the eyes. All these are examples on mechanical defenses that our innate immune system uses to protect us against infection. Now, if an infection occurs, these mechanical mechanisms may become overactivated. For example, in GI infections, we may see diarrhea and vomiting, which explosively expel pathogens out of our system. In respiratory infection, we may see increased mucus secretions, coughing, and sneezing, which also help expelling pathogens away from our body. Now, in addition to those, we have another physiologic barrier, which is the body's temperature. Our body's temperature is not suitable for many microbial pathogens, and even at physiologic temperatures, a lot of microbes can't survive. When the inflammatory response is initiated in the local tissues, cytokines may act systemically to alter the temperature set point in the hypothalamus, resulting in fever. The heat of fever acts as an innate barrier by inducing stress on pathogens and infected cells. Fever also augments the performance of immune cells and increases the efficiency of our adaptive immune responses. So these are the main barriers that our body uses in its first line of defense. And keep in mind that these barriers are actually interconnected in their functions and mechanisms and act together to combat pathogen invasion. For example, we said that skin is an extremely effective physical barrier due to its multiple epithelial layers and tight junctions between epithelial cells. But skin is actually unique because it has features of the other barriers as well. Skin is slightly acidic, so it maintains a chemical barrier which inhibits the growth of many microbes and potential pathogens. Skin also is inhabited by various microorganisms called skin flora that contributes to our defenses as a biological barrier. And finally, the shedding of dead skin and the perspiration of sweat plays an important role in the mechanical defenses that expel pathogens out of our body. Another example is mucus, which covers almost all the mucosal surfaces. We classified mucus as being a physical barrier which prevents pathogen contact with epithelial surfaces. But mucus is also unique because it has features of the other barriers as well. Mucus contains lots of antimicrobial proteins and peptides, and those actually maintain a chemical barrier against pathogens. Mucus also contains an ecosystem of commensal microbiota that contributes to our defenses as a biological barrier. And finally, the movement of mucus up the trachea via the mucociliary escalator or down the intestine via peristalsis is part of the mechanical defenses that expel pathogens out of our body.